Chromosomal Inheritance. Chromosomes. Chromosome is a structure found inside the nucleus of the eukaryotic cell. The DNA molecule is packed into thread-like structures called chromosomes. A chromosome is made up of proteins and DNA organized into genes. Each eukaryotic cell normally contains 23 pairs of chromosomes. Then, based on position of centromere, the 23 pairs of chromosomes in human are classified into 7 groups. They are Group A, Group B, Group C, Group D, Group E, Group F, and Group G. Group A. This group includes the largest chromosomes 1, 2, and 3, which are metacentric with centromere found in the center with 2 equal arms. Group B. This group represents chromosomes 4 and 5, which are submetacentric with 2 unequal arms. Group C. This is the largest group containing chromosomes 6, 7, 8, 9, 10, 11, 12, and X, which are of medium size and submetacentric. The X chromosome resembles the chromosome 6 in this group. Group D. This group includes chromosomes 13, 14, and 15, which are medium-sized chromosomes with nearly terminal centromeres. Group E. This group consists of chromosomes 16, 17, and 18, which are shorter, meta or submetacentric. Group F. This group represents chromosomes 19 and 20, which are shorter, metacentric. Group G. This group comprises chromosomes 21, 22, and Y, which are very short and acrocentric. History of chromosomes. Chromosomes were first described by Strasburger in 1815, and the term chromosome was first used by Waldeyer in 1888. Types of chromosomes. Chromosomes contain 2 types. They are sex chromosomes and autosomes. We already know humans have 23 pairs of chromosomes. Among these, 22 pairs are autosomes and remaining 1 pair is sex chromosomes. Sex chromosomes. Genetic traits that are linked to the sex of the person are passed on through the sex chromosomes. Sex chromosomes carry genes which specifically determine the sex. For example, XY is male and XX is female. Autosomes. Apart from sex chromosomes, rest of the genetic information is present in the autosomes. The autosomal chromosomes have thousands of our genes arranged on them, but these genes are not able to determine sex. Chromosomal mutation. Chromosomal mutation may cause permanent changes in chromosome structure. It can be caused by exposure to radiation, organic chemicals, viruses, or replication mistake. Only mutation in sex cells are passed on to the next generation. Structural changes in chromosomes. There are 4 major types of structural changes in chromosomes. They are inversion, translocation, deletion, duplication. Inversion. It occurs when a chromosome segment turns around 180 degrees. For example, hemophilia A. Translocation. It refers to the movement of chromosomal segments to another non-homologous chromosomes, for example, Down syndrome. Deletion. It occurs when a portion of the chromosome breaks off, for example, Turner syndrome. Duplication. When a portion of a chromosome repeats itself is duplication. For example, Pallister-Killian syndrome. Nondisjunction. Failure of paired chromosomes to separate during cell division so that both chromosomes go to one daughter cell and none go to the other is called nondisjunction. Nondisjunction causes errors in chromosome number such as trisomy 21 Down syndrome and monosomy X Turner syndrome. It is also a common cause of early spontaneous abortions. Example, Down syndrome. This abnormality was described by Down in 1866. It is caused by chromosomal aberrations. This is due to trisomy in 21st pair of autosome. Down syndrome is due to 47 chromosomes instead of 46. Chromosome number 21 is represented by 3 copies. Then, it is caused by autosomal aneuploidy. Hence, the chromosomal makeup is 21AA plus A plus XX. Symptoms of Down syndrome. The facial features of the individual resemble Mongolian race. The mouth is constantly open and the tongue is protruded. They are mentally retarded. The neck is short and broad. The female is dwarfed. The nose is oblique. The ears are malformed. This syndrome appears much more frequently in children born to women in the later part of the reproductive life. Sex determination. Differentiation as male and female sex and reproduction through sexual processes are basic phenomena in the living world. Sexual reproduction is a strategy for the production of diversity in nature. It is achieved through efficient function, anatomy of male and female individuals. In this natural system, the sex cells and the basic reproductive organs are called the primary sexual characters. Morphological, physiological, and behavioral characters exhibited by male and female of the species are called the secondary sexual characters. Thus, the characteristic difference between male and female animals constitutes sexual dimorphism. Sex determination in male. Example: Kleinfelter Syndrome. It is a sexual abnormality in males discovered first by Harry in 1942. Kleinfelter Syndrome is a genetical disease caused by an additional X chromosome in human male. It is caused by trisomy. Here, one chromosome is added to a set. This abnormality is due to the presence of 47 chromosomes instead of 46. The individuals possess an additional X chromosome with XY. So, the chromosomal makeup is 22AA plus XXY. Symptoms of Kleinfelter Syndrome. Some changes in Kleinfelter Syndrome person. They are sterile males. The testes are small. There is no spermatogenesis. Male sex glands are poorly developed. The breasts are enlarged. They are tall. Amount of male hormone is low and they are mentally affected. Sex determination in female. Example: Turner syndrome. It is a sexual abnormality in female discovered by Turner in 1938. Turner syndrome is a genetical disease in human female caused by the absence of one X chromosome. It is caused by a chromosomal aberration and also by monoploidy, where one chromosome is lost from one pair. This abnormality is due to 45 chromosomes instead of 46. The missing chromosome is one X chromosome. Hence, the chromosomal makeup is 22AA plus X0 is equal to 45. Symptoms of Turner syndrome. The baby develops into a sterile female because there is no menstruation but has female phenotypes. Ovaries are represented by a ridge of whitish tissue called streak gonad. Female hormones are low. The chest is broad, breasts are poorly developed, affected females are dwarf and mentally retarded. Inheritance. Inheritance is a process by which genetic information is passed from parent to child. This is why members of the same family tend to have similar characteristics. Inheritance is divided into sex-linked inheritance in Drosophila and sex-linked inheritance in human. Chromosomal inheritance. Most of the inheritable characters are controlled by genes located in autosomes. The genes that occur only on X chromosomes are called as X-linked genes. Similarly, the genes that occur only on Y chromosome are called as Y-linked genes. The inheritance of X or Y-linked genes is called sex-linked inheritance. First, The sex-linked inheritance were observed in Drosophila. Sex-linked inheritance in Drosophila. T.H. Morgan in 1910 studied on inheritance of genes in Drosophila and discovered that the pattern of inheritance of certain traits were found to vary with the sex of the parent and the offspring. He found that the genes for white eye color is X-linked. Further, it was found to be recessive to another X-linked dominant gene for red eye color. Red-eyed female into white-eyed male. When a wild red-eyed female Drosophila is crossed with a mutant white-eyed male, all the F1 individuals in both males and females have red eyes. When the red-eyed male and red-eyed female of the F1 were intercrossed, in the F2 generation, all the female flies were found to be red-eyed. Among the males, 50% had red eyes and another 50% had white eyes. White-eyed female into red-eyed male. When a white-eyed female Drosophila is crossed with a red-eyed male, all the female individuals in the F1 generation are red-eyed and all the males are white-eyed. When this red-eyed female individuals and white-eyed male individuals of F1 are intercrossed, the F2 generation possessed 50% red-eyed and 50% white-eyed females. Similarly, the male population of F2 included 50% red-eyed and 50% white-eyed flies. Sex-linked inheritance in human. Most of the sex-linked characters in human are X-linked. There are 150 confirmed X-linked traits known. Most of them are recessive. Based on sex chromosomal inheritance, they are classified into 7 patterns: autosomal dominant inheritance, autosomal recessive inheritance, X-linked dominant inheritance, X-linked recessive inheritance, Y-linked inheritance, codominant inheritance, and mitochondrial inheritance. Autosomal dominant inheritance. One mutated copy of the gene in each cell is sufficient for a person to be affected by an autosomal dominant disorder. In some cases, the condition may result from a new mutation in the gene and occur in people with no history of the disorder in their family. In others, an affected person inherits the condition from an affected parent, which results in 50% chance of having an affected child with one mutated gene, which is of dominant condition, and a 50% chance of having an unaffected child with 2 normal genes, which are of recessive condition. The most common inheritance is polycystic kidney disease. Polycystic kidney disease. It is an inherited disorder in which clusters of cysts develop primarily within kidneys. It causes kidney to enlarge and function over time. Cysts are non-cancerous round sacs containing fluid. The cysts vary in size and they can grow very large. Having many cysts or large cysts can damage kidneys. Symptoms of polycystic kidney disease. It includes high blood pressure, back or side pain, blood in urine, headaches, kidney stones, kidney failure, urinary tract or kidney infections. Autosomal recessive inheritance. In autosomal recessive inheritance, both copies of the gene in each cell have mutations. The parents of an individual with an autosomal recessive primary condition each carry one copy of the mutated gene, which results in 25% of unaffected son and 25% of affected daughter and 50% chance of carrier. Autosomal recessive disorders are typically not seen in every generation of an affected family. The most common inheritance is sickle cell anemia. Sickle cell anemia. It is a genetic syndrome caused by an autosomal mutant allele, hemoglobin S. In homozygous condition HbS, it causes the production of an abnormal hemoglobin called hemoglobin S. Normal hemoglobin is designated as hemoglobin A. Sickle cell persons with the genotype HbS suffer from a fatal hemolytic anemia. Symptoms of sickle cell disease. In heterozygous genotype, HbS-HbA are said to be carriers and they can survive. Homozygous condition can cause damaged heart, kidney, spleen and brain. Finally, it leads to death. X-linked dominant inheritance. X-linked dominant disorders are caused by mutations in the genes on the X chromosome, one of the two sex chromosomes in each cell. In females who have two X chromosomes, a mutation in one of the two copies of the gene in each cell is sufficient to cause the disorder. In males who have only one X chromosome, a mutation in the only copy of the gene in each cell causes the disorder. In most cases, males experience more severe symptoms of the disorder than females. In X-linked dominant inheritance, fathers cannot pass X-linked traits to their sons. No male-to-male transmission. The most common inheritance is Rett syndrome. Rett syndrome. Rett syndrome is a genetic disorder. Less than 1% of recorded cases are inherited or caused from one generation to the next. A few families have been described with more than one affected family members. This determines that the Rett syndrome has an X-linked dominant pattern of inheritance. A condition is considered X-linked if the mutated gene that causes the disorder is located on the X chromosome. The inheritance is dominant if one copy of the altered gene in each cell is sufficient to cause the condition. Symptoms of Rett syndrome. It causes abnormal pattern of respiration. There will be no speech development, mental retardation in childhood, loss of developmental milestone, high-pitched cry. X-linked recessive inheritance. X-linked recessive disorder is caused by mutations in genes on the X chromosome. In males who have only one X chromosome, one altered copy of the gene in each cell is sufficient to cause the condition. In females who have two X chromosomes, a mutation would have to occur in both copies of the gene to cause the disorder. Because it is unlikely that the females will have two altered copies of this gene, males are affected by X-linked recessive disorder much more frequently than females. In X-linked inheritance, the fathers cannot pass X-linked traits to their sons. No male-to-male transmission. The most common inheritance is hemophilia. Hemophilia. It is bleeding disorder that slows the blood clotting process. Major types of this condition are hemophilia A, which is also known as classic hemophilia or factor VIII deficiency, and hemophilia B, which is also known as Christmas disease or factor IX deficiency. Symptoms of hemophilia. In severe cases of hemophilia, continuous bleeding occurs in the joints, muscles, brain, or other internal organs. Y-linked inheritance. A condition is considered Y-linked if the mutated gene that causes the disorder is located on the Y chromosome, one of the two sex chromosomes in each of a male cell. Because only males have a Y chromosome, in Y-linked inheritance, a mutation can only be passed from father to son. The most common inheritance is Y chromosome infertility. Y chromosome infertility. This is a condition that affects the production of sperm and causes male infertility, which means it is difficult or impossible to pass the Y-linked chromosome as there will be no progeny. Symptoms of Y chromosome infertility. The affected man's body have less number of sperm count. In some cases, few sperms are present or unmatured sperm cells are present. Codominant inheritance. In codominant inheritance, two different versions, alleles, of a gene are expressed and each version makes a slightly different protein. Both alleles influence the genetic trait or determine the characteristics of the genetic condition. The most common inheritance is ABO blood group. ABO blood group. In codominance, two alleles are equally expressed in the phenotype of an organism. It can be easily seen in blood type. Group A blood type person depends on the type of antigen present in their red blood cells in the cell membrane. Type A blood has antigen A, type B blood has antigen B, type O blood has neither antigens. Both A and B are dominant while O is recessive. So, if a person with homozygous genes with type A blood has a child with someone who has type O blood, the child will have type A blood. However, if someone with homozygous type A blood has a child with someone who has homozygous type B blood, their child will have type AB blood. Both antigen A and antigen B will be present on the cells, and this is because A and B are codominant. Type O blood can be donated to any type of blood because it does not have A or B antigens. Type AB blood can only be donated to people with AB blood because it has both A and B antigens. Mitochondrial inheritance. Mitochondrial inheritance, also known as maternal inheritance, applies to genes in mitochondrial DNA. Mitochondria are structures in each cell that convert molecules into energy. Each contain a small amount of DNA. Because only egg cells contribute mitochondria to the developing embryo, only females can pass on the mitochondrial mutations to their children. Conditions resulting from mutation in mitochondrial DNA can appear in every generation of a family and can affect both males and females, but fathers do not pass these disorders to their daughters or sons. The most common inheritance is Leigh syndrome. Leigh syndrome. Leigh syndrome is an inherited neurometabolic disorder that affects the central nervous system. Mutation in mitochondrial DNA and over 30 genes in nuclear DNA have been implicated in Leigh disease. In the case of Leigh disease, crucial cells in the brainstem and basal ganglia are affected. Disorders of oxidative phosphorylation, the process by which cells produce their main energy source of adenosine triphosphate, may be caused by mutations in either mitochondrial DNA or nuclear encoded genes. The oxidative phosphorylation is most commonly disrupted in Leigh syndrome, either because of malformed protein or because of an error in the assembly of this complex. Symptoms of Leigh syndrome. It begins in infant and leading to death within a span of several years. Infants have diarrhea, vomiting, dysphagia, which means trouble swallowing or sucking, leading to a failure to thrive, hypotonia, which means low muscle tone and strength, dystonia, which means involuntary sustained muscle contraction, ataxia, which means lack of control over movement, are often seen in people with Leigh disease.